Okay, I think we'll get started with our second presentation of the day. This is going to be a research-based seminar, so please use the appropriate evaluation tool. All of the tools are posted in D2L and submit them to the Dropbox in D2L by midnight tonight. So with that, I am pleased to introduce David McWethy, who's going to speak to us on the effect of chronic methylphenidate treatment in an experimental mouse model of Parkinson's disease. So whenever you're ready.
Thank you, Dr. Mueller. And like she said, my name is David. This is a research we've been doing with Dr. Pond here. And we were looking at the effect of chronic methylphenidate treatment in the experimental mouse model of Parkinson's disease. And by the end of this seminar, I would hope, let's see if I can get it to move. At the end of this seminar, I would hope to be able to explain the toxicity of excess dopamine, describe the pathophysiology of Parkinson's disease, and analyze methylphenidate's contribution to oxidative stress. So first, we kind of going to go over a little bit of what we use methylphenidate for. So we use it for attention deficit hyperactivity disorder, and this is typically a childhood disorder that we use for like inattentiveness, hyperactivity, unable to talk successfully, unable to sit still. About 11% of children have been diagnosed with ADHD at some point, and 62% of those are currently receiving medication as a treatment option. And stimulants are the first line treatment for ADHD. And one of those stimulants is methylphenidate. In fact, that is the most commonly prescribed stimulant for the ADHD. Brand names are known as Ritalin, Metadate, Concerta, Falkland, Datrana. They're all a little bit different in their release mechanisms. They have different time frames of when they work. They're all indicated for ADHD and narcolepsy. However, there is some illicit use in these, using them for attention focusing, weight loss, and euphoria. A lot of studies have shown them to be most abused in college-age students really looking for the extra step to study. So how methylphenidate works is it blocks the dopamine transporter, which what that does, once dopamine is released, it takes up the dopamine into the presynaptic cell, kind of stopping its action in the postsynaptic neuron. And so this block keeps it from doing that. This methylphenidate blocks it, so it keeps it from doing that and allows for a greater response to a one dopamine release or one action potential release at the end of the presynaptic cell. It does work also on norepinephrine. However, it says there's much greater affinity for the dopamine transporter versus the norepinephrine transporter. So some adverse effects to this, to methylphenidate, is a weight loss, insomnia, motor tics, and there are also an abuse risk because you're working with the dopamine receptors. We will talk about dopamine for a little bit. So it's a neurotransmitter that has both central and peripheral neurological action. Centrally, it's important for mood, movement, reward, and attention. The attention is that big one while we use it for ADHD. Peripherally, it regulates heart rate and blood pressure. It can be used as a oppressor in case of other ones do not work. It interacts with dopamine receptors. There is a family of them. There's the intricacies of them is a little bit beyond the scope of the seminar. And again, like the action is halted by the presynaptic cell being taken up by the dopamine transporter. It also, some glial cells on the side will also pick them up. They are sequestered in the vesicles from there with the vesicular monoamine transporter into the seminal vesicles to be stored until the next action potential ends at the terminal and there's more dopamine release. It's either that or it's broken down by a monoamine oxidase enzyme or the catecholamine O-methyltransferase enzyme. So here are some of the major dopamine pathways. The one we're going to be looking at the most is the nigrostriatal pathway. This is mostly used for motor control, and the death of these neurons ends up in Parkinson's disease, which will be what we're looking at as the research looks at particularly. The other ones are mesolimbic and mesocortical pathways. Those are really used for memory, motivation, reward, and addiction. And they think the misregulation of these pathways may lead to schizophrenia. And there's one smaller one, the tuberoinfundibular pathway, which goes from hypothalamus to pituitary gland. It's kind of the connection between both the nervous system and the endocrine system, really responsible for hormone regulation and maternal behavior. So now methylphenidate, increases the dopamine levels in the synaptic cleft. So you think, great, we have better memory, better able attention span, feeling a little bit better about yourself. Unfortunately, excess dopamine does cause some neurotoxicity. And it does that because dopamine at physiological pH can self-oxidize into this dopamine quinone. And it can continue to do these chemical changes to form these free radicals down here, both as this compound, that's a direct substrate, and also these hydrogen peroxide as well. And these free radicals are very, very reactive. They will interact with anything and everything they can get their hands on and cause neurotoxicity. And the way that the cells keep this from happening is from glutathione conjugation. Glutathione is this molecule right here that kind of really helps bind to these potentially active molecules and shunts them off, either puts them in the cerebral spinal fluid or changes it into this neuromelanin. Particularly to note, the Niagara-striatal pathway, the reason why it's called the substantia nigra is because of this melanin. It causes a very deep color, very much like your skin. And that's just for this particular pathway that might be important. And so we look at Parkinson's disease now, which again is the neurodegenerative disease caused by the loss of dopamine neurons in the Niagara-striatal pathway. It has both motor and non-motor symptoms. Typically, motor symptoms start first. Usually, the biggest one you see initially is the symptomatic asymmetric resting tremor, and eventually it ends into this Parkinson-like gait, where they're very postural instability. They're very shuffle steps, very small steps, and they have this bouts of freezing where they can't cross barriers or they can't keep going forward in motion, even though they want to. They have that desire. They just can't seem to do it. Non-motor symptoms include depression, anxiety, cognitive impairment, and insomnia. My grandfather had Parkinson's disease, so I can really relate to some of these. He had a lot of cognitive issues later on as the disease progressed. Approximately 930,000 people live in the U.S. with Parkinson's in the U.S. Some notable people that you may have heard of is Michael J. Fox. He has a whole foundation looking for the cure for Parkinson's disease. Muhammad Ali, if you see some of his later videos as you age, you can see a very pronounced tremor in his hand. Also, George H.W. Bush recently passed away with Parkinson's disease. So we wanted to explore how does methylphenidate increases the dopamine release, and but also that can lead to oxidative stress. And we wanted to see how that long-term use of methylphenidate could potentially cause this dopamine neuron loss. And so what we did was the research question that we looked at was, does methylphenidate sensitize once resistant dopamine neurons to the Parkinsonian toxin MPTP? And MPTP stands for 1-methyl-4-phenyl-1,2,3,6 tetrahydropyridine. I'm going to call it MPTP from now on. That's a mouthful. And what they found is a contaminant found in MPPP, if it was not synthesized correctly. MPPP was a Demerol. They're searching for a new alternative to Demerol, maybe less abuse potential. And this is one of the compounds that the pharmaceutical company found. They never proceeded because they didn't find it wasn't a great alternative. However, they did publish papers citing how to make this. And some industrious people found these papers and thought, hey, there's this compound out there that acts like Demerol, but yeah, it's not scheduled on the FDA, so it's legal to make. So first, a 23-year-old grad student made this stuff, and he did have bouts of. At one time, he injected it, and he got a burning sensation in his arm, and he went to the ER, and they found it very symptomatic, like Parkinson's disease. Now, Parkinson's disease is very much an older person's disease. So a 23-year-old getting Parkinson's like symptoms is very rare and very doesn't happen. And so they never figured out why. They did a case report on it. They kind of just went away and couldn't figure out why this particularly happened to the patient. It wasn't until later where a bunch of heroin addicts came in with the same, very similar Parkinson's disease after they injected this new heroin. And what they found was someone had found this MPPP, tried to start to make it, and got this contaminant of MPTP. And so these people were injecting it and got Parkinson's disease from the dopamine cell loss that happened afterwards. And MPPP causes this dopamine cell loss by this mechanism, which I'll explain here in a sec. So MPTP in itself isn't neurotoxic. It crosses the blood-brain barrier just fine, doesn't do anything while it's in there. What happens, it gets taken up into the glial cells, gets enzymatically changed by monoamine oxidase B, and gets changed as MPP+. Now this is where the toxicity comes in. MPP+, while it doesn't look exactly like dopamine, dopamine transporter readily takes it up. So it gets into those dopamine neurons just fine. And once it's there, it can interact with the mitochondria and decouple the ATP production with the phosphorylation. It causes a lot of mitochondria death and a lot of releases a lot of oxidative stresses. Also of note, just like the dopamine transporter, this vesicular monoamine transporter, what sequesters the dopamine into vesicles, really enjoys MPP plus as well. And that means that these MPP will actually sit in these vesicles and wait till the next release, and then they can do their oxidative stress all over again. So we see a very long-lasting cell loss, and what this is looking at is MPTP-induced cell death in these mice C57 black 6 mice, which are very susceptible to this MPTP toxin. And even after just one dose of MPTP or 1 days of injection, they find significant cell loss even at day 7. So this is a very long-lasting effect, and so it's just and that kind of is explained by the vesicles being able to store it until for later use. So this is actually Dr. Pond's postdoctorate work. She looked at different mice strains and how they react to MPTP, and they found one particular mouse strain called the Swiss Webster mice that were very resistant to MPTP. So right here you can see that those would give them control, and MPTP saw no significant decrease in dopamine neuron counts. Even after all this oxidative stress, they had very resistant to this toxin. However, once they introduced this methylphenidate, they started to see significant losses in dopamine counts. So they think that maybe the oxidative stress of methylphenidate might have added, prevent, and reduce some of the resistance that they initially had. And they've come to find out they also gave these mice 10 milligrams per kilogram of methylphenidate and found significant decrease in dopamine levels, and just that alone. It doesn't, not even with any MPTP. Just that alone was enough of an oxidative stress to lose neurons, and which again was even greater loss when they induced the MPTP in those mice. So we have this great data on this dopamine neuron counts. However, we wanted to look at more of the biochemistry of this, and so we came up with the idea of looking at the chronic methylphenidate induces increased quinone production and subsequent depletion of antioxidant glutathione in the striatum. And this is the project that I kind of came in on. And so what we did, this is our methods. We took male Swiss Webster mice and injected them intraperitoneal twice daily with starting on P28 with saline, 1 milligram per kilogram methylphenidate, or 10 milligrams per kilogram methylphenidate. We chose it twice daily because it's an immediate release drug. We can't really have a long-lasting effect from an injection because of the way the drug works. So we did, and that's what you see if you're giving the methylphenidate that's not extended release versions of it. They're usually given twice a day. We picked the P28 because that is the 28th day after birth of these mice, and that usually relates to about pre-adolescence in human children. That's the kind of on their lifespan. That's where we're at. And then we use 1 milligram per kilogram of methylphenidate for the therapeutic use of methylphenidate for ADHD. We selected the 10 milligram per kilogram for abuse use of methylphenidate. So just increased dosing for to kind of stimulate the abuse of methylphenidate use. So the mice receive injections Monday through Friday for 12 weeks with a 7-day washout. And the Monday through Friday was to stimulate the school week. In 12 weeks, that ends up mice being about, like, in human terms, about in their 10 years. So late teens, early 20s. And we gave them a 7-day washout because, like, if you remember, so MPP plus, the toxin, gets drawn up into the cell with the dopamine transporter. Methylphenidate blocks the dopamine transporter from being able to take up dopamine. So we didn't want this protective effect that methylphenidate may induce in these mice, because the MPP can't be taken up by the dopamine cells before it has its chance to exert its effect. So we had to do a 7-day washout to make sure there's no methylphenidate left in these mice. And then we gave them their injections of either MPTP, 4 doses at 20 milligrams per kilogram every 2 hours, or of sterile saline. And again, we separate them into each. So here are some results. That we're looking here is the quinone production. Again, quinone is what happens when dopamine is in excess and allowed to oxidize. So we see a significant increase in dopamine in quinone production with a 1 milligram per kilogram of methylphenidate, and even greater increase with the 10 milligrams per kilogram of methylphenidate. And this really speaks to how much excess dopamine is in the synaptic cleft with the addition of methylphenidate, even after a 7-day washout period. So like we said, that these dopamine quinones are kind of a surrogate for oxidative stressors that we can't really, unfortunately, see very clearly, can't really test very clearly. So we use these as kind of a surrogate. So these are upstream from those oxidative stressors. So it really shows how much oxidative stress could potentially be on these neurons with methylphenidate at these doses. Interestingly enough, with the addition of MPTP, we didn't see that increase that we saw in the others. And what we kind of thought was maybe the issue here is that with the MPTP, we saw a significant amount of cell death. And so without the cells there to make the dopamine, there wasn't enough dopamine to release to make the quinones. So that's what we kind of assume happened. We don't have a really great indicator of any other options. So now we're going to be looking at the glutathione levels. Again, the glutathione is what conjugates these molecules to prevent them from going on to be these free radicals and oxidative stressors. We didn't see any significant difference between the saline and the 1 milligram per kilogram of methylphenidate. However, when we got to the abuse dosing that we used, the 10 milligrams per kilogram of methylphenidate, we did see a significant decrease in glutathione levels in both the ones without MPTP and even a greater increase in ones with MPTP. Just kind of again shows how much oxidative stress at this dosing these mice the neurons had to undergo, and how much glutathione was being used to kind of prevent the extra damage from these oxidative stressors. So long-term, the conclusion of this, long-term exposure to methylphenidate increases dopamine quinone levels while depleting available glutathione. This may lead to decreased resistance to oxidative stress that may damage dopamine neurons and pathways. So now we have all this great data on the male mice. Perfect, but unfortunately we didn't have any great data on the female mice, and so females are not the same as males. We'll see that shortly. They have different levels of dopamine transporters and things of that nature. So what we want to look at is the effects of chronic methylphenidate use in female model of Parkinson's. We use a very similar methods. A lot of the same time frame with starting injections, the same concentrations of methylphenidate. We did separate them in the female mice into 2 additional groups, one an anestrus and proestrus. And what that is so mice are unique compared to us, where if they're in without males, so they don't have any male input, male hormone, pheromones, they will go into an anestrus state where they're no longer reproductive viable until they receive that stimulation of a male pheromone again. So what we did was we separated them into 2 groups. We injected them all while they're all in anestrus states. We did not give them any male pheromones to induce the estrogen cycle during methylphenidate use. However, on the last day of methylphenidate use, we did place some male pheromones into the proestrus cages in the form of male urine, and we confirmed that they're in an estrogen state with a vaginal spear on the day of MPTP injections. So the eagle eye viewer may have noticed that we used a little bit different MPTP dose for the female mice than we used for the male mice. At the end, we used a 12.5 milligram per kilogram of MPTP in the female mice, when we used a 20 milligram per kilogram of MPTP in the male mice. The reason we had to do this is when we tested this to make sure that this experiment was viable, we used the 20 milligram per kilogram in the female mice, and they all died before we were able to get samples off of them. So we already understand different mouse strains react differently to this toxin, and obviously different genders will react different to this toxin as well. And there's no great study to show us how Swiss lobster female mice react to MPTP toxin. So it was very much a surprise when we used this. We just kind of proof of concept, and we didn't have any samples. So we find that females have a higher peripheral toxicity compared to males to this toxin. So we did a little study trying to find what may be the best concentration of MPTP to use in these mice, considering that we still want to see an effect. However, we do need samples at the end of this study. So we settled on the 12.5 milligram per kilogram, which is about 50% probability of survival. Unfortunately, this means that we can't directly relate this to the male model because of the different dosing of MPTP. It wouldn't be statistically sound. So here are some results. This is on the left here, you'll see pictures of the striatum. These dark spots are neurons that have been dyed with tyrosine hydroxylase. And that's an enzyme that's very important to forming dopamine. That's kind of how we could tell which ones were dopamine neurons, which ones were not. And you can see on the right, after MPTP use, you can see it's lighter. It's a little bit lighter, especially in the 10 milligram per kilogram of methylphenidate. However, when we go to the broader spectrum of the data, we see that we couldn't find very much significance between the two. The only place we found significance is in the saline versus in MPTP versus no MPTP. We found a significant decrease in neuron counts. I would like to note that while we do see a trend, but these confidence intervals are fairly wide, and I think that's what limits us to seeing statistical significance here. And in the estrus females, we found no statistical significance, and that's going to be a recurring theme as we look at these results. So now we looked at the quinone production of these females, and we saw in the pro-estrus females, there was a significant increase in quinone production in the MPTP. However, we did not find the same with those treated with methylphenidate. We actually found a significant decrease compared to that of the saline and MPTP. And what may have happened in this case is MPTP in itself is a retrograde toxin. It starts in the axon terminals of the cell and slowly works up towards the cell bodies. And while we did not see any cell death as a dopamine neuron cell bodies death, the terminals may have been destroyed enough where there was a decreased release of dopamine and thus a decreased ability to make dopamine quinones. That's a potential explanation. We have to redo the study and look at possibly more data to try to figure out if that may be the case. But that's what we kind of came up with. There's no significance in the anestrus female, again, most likely because of the wide intervals that we have. So we also looked, again, just like the males, we looked at the glutathione levels. And here's where the bulk of our statistical significant data we found. So we found a significant decrease in glutathione levels in both the ones treated with methylphenidate and the ones that treated with MPTP. And so we can see that even without all of this quinone productions and the like, we do see a significant decrease in glutathione levels, kind of a correlation of increase of oxidative stresses on these mice at this time. And we do see a significant decrease in glutathione levels compared from the 10 milligram per kilogram of methylphenidate saline MPTP versus non-MPTP. And again, this speaks to more of how much resistance is lost with the introduction of this large amount of methylphenidate in these mice. And this is also the only time we found anestrus significance, where those treated with 10 milligrams per kilogram of methylphenidate had significant decrease in glutathione levels compared when they were given MPTP. So our conclusions we got from here is that estrogen seemed to sensitize dopamine neurons to MPTP, as pro-estrus animals experience greater cell loss and quinone formation following MPTP without methylphenidate. And it seemed long-term exposure to methylphenidate led to depletion in available glutathione levels. And again, methylphenidate may decrease resistance to oxidative stress that may damage neurons and neuronal pathways. So this is all great. But how does this translate to humans? Most of us are pharmacists. We treat humans. Very few of us will go on to be veterinary pharmacists. So I wanted to see, how does this relate to our humans? I do want to mention that I named this mouse personally Pippa after my dog, because she just loved to bite me every chance she got. So I just wanted to do a shout out to Pippa. So I was looking at potential studies to look at this correlation between ADHD and Parkinson's disease in the human population. And I found this observational study. It looked at the increased risk of disease of the basal ganglia and the cerebellum in patients with a history of ADHD. So this study looked at, was a retrospective cohort study that was conducted on the Utah Patient Database. Now, a database is a large database of medical records from patients seen in Utah specifically. I believe it has about 85% of any patient seen by a medical professional. And so it's a very, very large group of people to look at. And so after this initial inclusion and exclusion criteria, they were able to pull, the source population was like 1.92 million individuals. Again, a very, very large population base. After they searched for ADHD patients, they were able to find 300,769 patients to put in their ADHD cohort. And they want to do a 5-to-1 ratio from non-ADHD cohort to ADHD cohort. So they picked 158,790 patients without ADHD to put into their non-ADHD cohort. And here are the results they found from that study. So they found that patients that had ADHD diagnosis were 2 to 3 times more likely to be diagnosed with the basal ganglia and cerebellar disorder later in life. I should have mentioned one such basal ganglia and cerebellar disorder is Parkinson's disease. They have other ones included in there, but that is one of the bigger ones that they were looking at. And they also found that patients with ADHD who were given stimulants were actually 6 times more likely to form basal ganglia disorders. And we can see here that this confidence interval from the 3 to the 3.9 did the giant intersect. So this is a significant difference between those given not given stimulants and those given stimulants. I would like to mention that they did look at methylphenidate as just one stimulant, because again, it is one of the more commonly prescribed stimulants given to ADHD patients. They couldn't find significance in that patient population, and we can see here this confidence interval is pretty wide. It goes from 15.1 to 4.2. There weren't a whole lot of patients with methylphenidate. I think they ended up with about 1,000 patients who were prescribed methylphenidate taking that. So the power there isn't the greatest. So the authors concluded that ADHD diagnosis may increase the risk of developing basal ganglia and cerebellar disorders in the future, and stimulant treatment may increase these risks in these patients. While I agree with the authors that there's a correlation between ADHD and the basal ganglia and cerebellar disorders in this population, again, this is a Utah population. I believe about a little under 90% of the people that they looked at were Caucasian. So it kind of affects the external validity of this study. The stimulant use that they quoted was an increased risk. That one's a little bit more suspect. While stimulants may have been the cause for this increased risk, actually there can be no cause taken because this is a correlation study. The stimulants might be just more of an indication that their ADHD might be a more severe case, therefore they needed stimulants to handle their symptoms. They also didn't cover other confounders like head trauma or environmental toxins, of which actually NBTP, while not an environmental toxin, it has very similar structures to some pesticides that have been used. And once they found out that MPTP has this effect of Parkinson, of oxidative stress on dopamine neurons, they're worried that these pesticides that may have this similar effects. So in summary, methylphenidate appears to decrease Swiss-Webster mouse resistance to oxidative stressors. Patients diagnosed with ADHD are more likely to be diagnosed with a basal ganglia and cerebellar disorder later in life, which is increased even greater when treated with stimulants. While this causation of this finding is unknown, because this again was more of a correlation study, there was no actual causation can be pulled from this. Our research kind of gives a biological mechanism of how this potentially could be if it is in fact the stimulants causing this increased risk. And that kind of is a clinical conclusion I want to put in here. So stimulants potentially have these long-lasting effects that not have been thoroughly studied or evaluated. And so I believe other treatment options should definitely be explored before trying stimulants, one of which is behavioral. In these patients, behavioral treatment is Honestly, it should be the go-to in these patients to try to help them work through the disorder. And if stimulants were used, I believe the lowest effective dose for the shortest duration should be utilized. And with that, are there any questions?
Questions for David?
David, you mentioned in some of your research that you had some wide confidence intervals in what you found. What's your sample size for one of these experiments?
Our sample size, they varied a little bit. They were from 5 to 10, and even in some of those, I'm sorry, the question was, what was my sample size for some of these experiments? We want to limit the amount of mice lives that we use, because again, these are being sacrificed, and so we limited it to about 5. I think the greatest number was 10 in one of these groups, and even after that, there was some mice death before we could get samples off of them. So it was smaller sample size, more of the fact that we wanted to limit how many mice we use in this study.
And then, is there any theory or any idea why there is such differences between male and female Swiss Webster mice?
So the question was, is there any theories of why there might be difference? And honestly, I tried to look that up, and there isn't a whole lot of data looking at that particularly. And honestly, there's just not a whole lot of studies looking at that particular question. It's a question I have as well.
Other questions?
I don't have a question, but I think you did a great job explaining this. I know when I joined the lab, Dr. Pond had to explain it to me about 5 times, so I don't know if it's just I already know what's going on, but I think you did a really good job breaking down the why behind our processes.
So where do you go from here with this information?
Yeah, that is a good question. And I've been thinking about that a lot. How am I going to use, the question was, where do I go from here from this? I've been thinking about it a lot, and really it's more of because anecdotally, I've seen a lot of older people be taking stimulants kind of help with attention, and they feel like they need to get things done, and that's how they can get through it. And I see that, and I don't agree with it. Now I have a better feel like, hey, these have long-lasting effects that could potentially harm you in the long run, and maybe you should just back off in some of your tasks that you've been trying to take on. Otherwise, for particular experiments, I'd be interested in seeing if these mice, the Swiss Webster mice, have that resistance if it comes back after long enough absence of methylphenidate or any stimulant in general. Because that'd be interesting to see that maybe potentially stopping methylphenidate in human population, do you return it to baseline, or is that damage already done? And that'd be an interesting thing to kind of see.
Thank you. Do you think there are any differences related to age? Because these were like adolescent mice, correct?
Yes. The question was, do I think there's any difference related to age? Honestly, I don't think, well, there could be potentially older mice may have less resistance to oxidative stress. just because they've been around longer. These neurons have been affected by stressors, because to see dopamine itself can cause oxidative stress if there's an excess. And just because we're stimulating a higher concentration doesn't mean that doesn't already happen. So it would be interesting to see like how the older population is affected in this. Is it affected the same way? Is it more drastic change? Or I personally, my hypothesis would be a more drastic change than even just adolescents.
Thank you. Other questions?
Hey DJ, I have a follow-up question from what Dr. Bazar asked about the effect of gender on toxicity. I know you saw that effect in mice, but do you think that that would affect any recommendations you would make in humans?
So the question was, we saw an increased toxicity in mice. Will that change my recommendation in humans? From what I understand, females actually have a higher concentration of methylphenidate in their brain as opposed to males. Supposedly they also have more dopamine transporters, which may cause a greater risk for the oxidative stress to happen, as there's more methylphenidate in the area, as well as more places for it to be gummed up.
Okay, I got a follow-up question then based on that. So I think in one of your slides you said that pro-estrus mice, that in them you said that estrogen sensitized their dopamine neurons. Is that really one of your slides said?
Yes. We assume that estrogen may have a protective quality.
Oh, you assumed.
Yes, that was our hypothesis. Our question was, why did estrogen seem to sensitize when? That was our hypothesis. Was we know estrogen to be a neuroprotective agent. However, when we studied it in these mice population, we found that actually was not the case in this particular. They actually sensitized it to this methylphenidate and NPTP toxin.
Could there be differences in, like you said, in humans, differences in how estrogen affects dopamine concentrations? So could it just be a concentration dependent effect that somehow higher estrogen levels somehow lead to higher dopamine levels in certain neurons?
Yeah, it definitely could be that. It's hard to say. Dopamine isn't the easiest thing to pull out of tissue. It's very difficult. That's why we didn't directly look at the dopamine levels. That's unfortunately a limitation on our study, as there's no great way of pulling out tissues.
Any other questions? All right, then we'll move into the feedback portion. So what comments does everybody have for David? What went well? Little tweaks to make it, improve it? Any suggestions?
I just wanted to say I think you did a really great job. I've seen you present your research, I think, twice now, and I think you did a great job, especially explaining like the background information. I know this is a really tough topic to really get into and explain, and like you did a great job of explaining the different pathways and neurotransmitters, and then also your research, your methods were very clear. So great job.
Thank you.
Other comments?
Well, I will will go. DJ, I thought you did a great job. It is a really tough, complex topic, as Caitlin mentioned. I mean, there's just so much complexity to it. And then, you know, the female data in particular wasn't exactly what we expected to see and sort of hard to explain for even me. And I'm immersed in this. So, so great job. I thought you did a really nice job. There is sometimes, and it could be the Zoom and the microphone and stuff, but sometimes it sounded like you mumbled a little bit. It was sort of hard to hear. So be careful with that enunciation. But overall, I really appreciated your presentation. I liked that you talked about your grandfather, added that personal touch. Your slides were great, like lots of pictures, and you took time to explain the pictures. The one thing I would do is kind of continue that, like when you're discussing your data, try, and I forget this too, but try to these are the axes to the graphs. This is the colors that we're looking at, and really orient the audience to the figure that way. I realized, you know, there's time constraint, and that might have been part of the issue today, but that's something that, you know, even like I said, I forget, but is really helpful when the audience is looking at that and trying to follow what you're saying. It kind of keeps everything together. So, but great job. I'm really proud of you.
Thank you, Dr. Pond.
I thought you did a nice job too. I appreciate, this has been so far removed from my brain because I'm old. But I could follow you very clearly, and you had really nice explanations in the background, and I think it led into your studies really, really well. And I like at the end how you, which is what we want you to do, even when you present your own personal research, is bringing it back to a practicing clinician and what this could possibly mean going forward. So nice job with that. And I had made a similar comment about orienting to charts. So yeah, that's an easy tweak that you can change going forward. Any other thoughts, comments? Well, if not, I think everybody's kind of zoomed out here. So this is hour 5. I love Wednesdays. So with that, if you do have any other feedback, just feel free to, you know, email our students here and give them that feedback. It's always very helpful going forward, because these presentations could be used for residency interviews or fellowship or job related. So any feedback is helpful. So with that, again, I remind all the students to upload their evaluations to the Dropbox in D2L, and faculty, if you could just email me your evaluation sheets as soon as you are able, and I will get these finalized, the grades. Thanks for attending, everybody. We'll see you.